Dementia Researcher with a blog and a rating.
The Blot thickens, using Western blotting in research. Western blotting has a bit of a reputation in the academic circles for being, well, temperamental, unforgiving, and witchcraft at its finest. Us researchers that have been lucky enough to run one, or many, of these bad boys, don't have the best memories of this technique. We often joke about making sacrifices to the Western gods, or that the Western will only work if the tank faces northeast. But, jokes aside, in this blog I will describe the basis of what a Western is, what it can tell us scientifically, and how I have used it in my own work in the lab. Western blotting, or immunoblotting, is a laboratory technique which is used to detect a specific protein or proteins within the complex mix derived from cells or tissues. Westerns can help us understand how much or how little a protein is expressed, or in other words, how abundant it is. Determine if proteins are being up or down regulated, detect cellular location of proteins, as well as giving us an understanding of the size of the protein, and if any of these measures might change, for example, in disease or after a treatment. But before we even get near the Western blot tank, the first step is deciding what samples we're going to run. Some tissue samples, like the brain, include many different proteins that are all expressed at different levels. Some proteins have lower expression and thus may be trickier to detect. In my work, I was particularly interested in proteins that make up the blood vessels of the brain, and to understand how the composition of these proteins may change in small vessel disease. Blood vessel and basement membrane proteins are notoriously difficult to detect. So to give myself the best chance of seeing these proteins, I developed a protocol to enrich my brain samples for blood vessel-specific proteins. My new brain sample then included many more vessels per volume than if I compared it back to my original whole brain sample. So now that we have our sample, we need to decide what type of Westerns we want to run. This is determined by what proteins we want to detect and how we can access them with antibodies. In a denaturing Western, it sounds painful, I promise it isn't. We boil our sample in a special buffer, known as anionic detergent sodium dodecyl sulfate, SDS for short, to break the structure of the protein. We do this because antibodies typically only recognize a small portion of the protein of interest, i.e. the epitope, and the epitope may reside within the 3D structure of the protein, which would be inaccessible unless we unfold it or denature it. For some proteins, this is not necessary, in which case we wouldn't denature a sample at all, which occurs in a native Western. After our sample preparation, we are ready to embark on the first step of Western blotting. Pause for drumroll. Gel electrophoresis. To put it simply, this technique separates the sample complex protein either by charge, native Western, or by weight, in a denaturing Western. If we denature that sample, as described above, the SDS buffer allows all proteins to become negatively charged as they attach to the SDS anions. The negative charge is proportionate to the length of the proteins, therefore migration through the length of the gel is determined by molecular weight. In other words, heavy proteins will run slowly and stay at the top of the gel, and small proteins will run quickly and end up at the bottom of the gel. This step is achieved by loading our sample into a well of a gel in a special Western blotting tank. Then when an electric current is applied to the gel, the negatively charged proteins will migrate towards the positive charge at the bottom of the gel and separate by size. The end result, hopefully, is a gel with lots of sequential lines on it. Sounds pretty straightforward, right? But lots can go wrong at this stage. If the current or buffer isn't right, this can lead to smiling or frowning lines, and depending on how wrong this is, it can lead to all different shapes. Twitter is flooded by researchers sharing their Westerns gone wrong, and so far the best shape I've ever seen must be this bowl, courtesy of Dr. Sophie Quick. Once the proteins have run and separated, they are transferred out of the gel to the surface of a membrane. This transfer of protein can happen in a couple of different ways, and will be determined by what size proteins you're interested in. All transfer types follow the same principle of the sandwich, which ironically happens around lunch, where the gel and the membrane are sandwiched together with blotting paper and sponges. An electric current is then applied, causing the proteins to migrate out of the gel and attach to the membrane. Needless to say that the order of the sandwich is crucial. If not assembled correctly, the proteins will either transfer onto the blotting paper or simply flow away into the transfer buffer. Bubbles aren't your friends either. They can cover your proteins, making it impossible to quantify your protein expression at the very end. Yes, remember when I said that Western blotting is not forgiving? I wasn't joking. Once the transfer has taken place, we can then take a breather and set the tank aside. A protein-full membrane is blocked to prevent non-specific binding from loose proteins that may have occurred during the transfer, and then probed with specific antibodies that bind to the target proteins of interest. This binding is then detected with a chromogen or a fluorescent tag. And finally, the membrane is imaged to reveal the expression or lack of the protein or proteins of interest. This data can then be quantified, where the intensity of each band can be measured and normalized to a control protein, thus measuring protein expression for each sample. In my own work, I have used Westerns for a couple of different reasons. Remember my blood vessel-enriched samples that I mentioned previously? With these samples, I was able to quantify expression of 4 key basement membrane proteins, where I was comparing across control mice and mice with a collagen 4 mutation, the same human mutation that leads to a form of genetic small vessel disease. I was able to measure the expression of these proteins and observed different levels of them in control and diseased mice, and that this expression pattern changed across brain regions. I've also used Western blots as a confirmatory tool for another type of brain enrichment, and then sent these human and mouse samples for proteomic analysis, i.e. the master technique for large-scale protein study. Away from the lab, Western blotting has also been used in a clinical setting to aid in the diagnosis of infectious diseases including HIV, Lyme disease, hepatitis C, syphilis, as well as some autoimmune disorders. Although it's gradually been replaced by the arrival of new, more sensitive and specific diagnostic assays that have been developed for some diseases. Thus, Western blotting remains a predominantly research rather than clinical tool. Despite the challenges, and yes, the frustrations that Western blotting can bring, it is still an invaluable tool in research to study and detect proteins, and also has applications in the diagnostic settings. And just remember to always make sure to turn your Western tank to face northeast.
Thank you for listening. Join the Dementia Research bloggers and share your own views.